So my name is Véronique Carsten, and as a product manager for molecular biology products for MP Biomedicals, I'm very pleased to give this presentation today, exploring the world of microbiomes with insights into state-of-the-art solutions for metagenomic approaches. So our agenda for today is to talk about microbiome research and sample preparation. I will start the presentation with a big picture of microbiome research applications and the common workflow of microbiome research experiments. This first step of the workflow is sample disruption and cell lysis, and this gives me the opportunity to introduce the FastPrep system, a complete and ready-to-use solution, ensuring a successful lysis of any microorganism. The next section of the presentation is dedicated to the FastDNA spin kit for soil, designed for the isolation of DNA from samples used in microbiome research. Several case studies are pointing out that this method is a gold standard for this application. And this encouraged MP Biomedicals R&D team to develop additional kits for targeted sample types like water samples and to come up with automated purification solutions that will be the focus of the last part of this presentation. Microorganisms building a microbiome are composed of bacteria, fungi, protozoa, archaea, and viruses. These fascinating microbial environments are found everywhere, thriving in virtually all parts of the Earth's biosphere. Major environments hosting microbiome communities are human and animal bodies, soil, oceans, and plant specimens. Research on microbiomes shows that these microorganisms are intrinsic to our health, and microbiomes are also fundamental to key geochemical processes. Human microbiomes synthesize vitamins and neurochemicals. They aid digestion, strengthen the immune system, influence processes in the brain and mental health, and play a role in certain cancers. Soil microbiome is involved in the cycling of nutrients and provide plant protection against biotic and abiotic stress. Ocean microbial inhabitant, also called nanobiome, produce half of the oxygen we breathe, and recycle the same proportion of carbon dioxide from the atmosphere. In plant, rhizobacteria play a role in plant growth, nutrient absorption, and disease resistance. Microbial communities are also used effectively to consume and break down environmental pollutants. They are involved in the digestion of plastics, removal of radiation, and cleaning of oil spills. Microbiomes are actually complex and dynamic ecosystems. Each microorganism has a specific role in the community and interacts with the host. The composition of microbial communities are known to change in response to external factors, factors such as diet or the climate, for example. Microbiomes can therefore be modified to maintain a beneficial effect, to improve their function, or to restore them when they are altered. This is why understanding microbiomes is essential, and research studies focus on studying the diversity and function of microbes in their own environment, investigating factors affecting the composition and function of a microbiome, and uncovering the role microbiomes play in human, animal, and plant health, as well as in the environment. Several technologies are used to explore microbiomes. The two major approaches for analyzing microbiomes are the 16S rRNA gene sequencing technique that allows the identification and quantitation of microorganisms, and shotgun metagenomic analysis that is conducted to enhance the knowledge on the taxonomic and functional profile of a microbial community. In parallel, metatranscriptionic allows to obtain whole gene expression profiling of complex microbial communities. These molecular biology technologies always start with the same experimental workflow: sample collection, followed by sample disruption and cell lysis that allows the subsequent isolation of DNA for 16S rRNA diversity analysis or metagenomic analysis. Microbiome research projects require an expertise in dealing with challenging samples, and sample preparation is a critical point. An inefficient method can have a big impact on the microbial community analysis and lead to biased results. An insufficient sample grinding step will affect the lysis efficiency of microorganisms included in the sample and therefore skew results. As well, an inefficient cell lysis procedure that selectively breaks open easy-to-lyse microorganisms, while difficult-to-lyse cells like spores and gram-positive bacteria, for example, will not be lysed, will affect the results of the microbial community analysis. As well, sample prep methods leading to low yields of DNA or poor DNA quality, or co-purifying substances coming from the sample like nucleases, proteases, or PCR inhibitor factors like humic acids included in soil sample, or phenol compounds and polysaccharides from plant samples will affect downstream experiments. With more than 20 years experience in developing comprehensive sample preparation solutions, MP Biomedicals offers an efficient method for the preparation of high-quality DNA from challenging sources, a guarantee for reliable downstream analysis. The FastPrep system includes several bead beating homogenizer for a thorough homogenization and cell lysis of the most resistant samples. They are used in combination with Lysing Matrix tubes containing a mix of beads of different material, size, and shape, optimized to guarantee a complete homogenization of the sample and the lysis of any microorganism. In order to provide a complete sample prep solution, ready-to-use purification kits containing the Lysing Matrix tubes for the grinding step and all the reagents required for the purification of DNA or RNA have been developed. 4 bead beating homogenizers have been developed to answer the need of specific application. On this slide, you can see the FastPrep 24 and FastPrep 24 5G instruments for middle throughput experiments performed in tubes of 2 ml, 4.5 ml, 15 ml, or 50 ml with the option of using a cryogenic sample holder to prevent a temperature increase of the tubes. The FastPrep 96 instrument that is specifically designed for high throughput application and screening experiments. 2 96 well plates can be processed simultaneously. And the mobile Super FastPrep 2 device is a convenient way to grind sample on site. FastPrep instruments are used in combination with Lysing Matrix E-tubes containing a mix of 0.1 silica beads, 1.4 mm ceramic beads, and 1 4 mm glass bead that are designed to grind efficiently any starting sample used for microbiome research applications. Lysing Matrix E is supplied in tubes of 2 ml, 4.5 ml, 15 and 50 ml tubes, as well as 96 well plates. The use of these tubes ensure the optimal and reproducible grinding and lysis of any microorganism included in the starting sample in just a few seconds. They are commonly used with FastPrep instruments, but are also compatible with any other bead beating instrument. The FastPrep system offers a straightforward, powerful sample prep procedure based on the unique figure-8 motion of the instrument, combined with performant Lysing Matrix particles designed to grind the most resistant samples. In order to purify DNA after the grinding and cell lysis step, MP Biomedicals developed a comprehensive range of complete DNA isolation kits. These kits are split into 3 main groups, depending on the purification protocol used to obtain pure DNA. fast DNA spin kits are based on a protocol purifying DNA with a DNA binding matrix in slurry, combined with spin filters used to facilitate the purification process. Spin-easy DNA kits include ready-to-use silica membrane spin columns designed for the purification of DNA. A complete range of specialized sample type-specific DNA extraction kits have been developed. And MacBeads DNA kits that are designed for automated purification of genomic DNA using DNA binding magnetic beads and following a bind-wash-elute process. The first kits MP Biomedicals launched were fast DNA spin kits, and one of them is today recognized as a gold standard method for DNA isolation from environmental samples. We will start with an overview of these specific kits. Fast DNA spin kits include a DNA binding matrix solution supplied in slurry that offers the flexibility to increase its volume when using cell-rich specimens and thus purify high yields of DNA. These kits include as well specialized Lysing Matrix tubes that guarantee a thorough homogenization of the most resistant samples and the lysis of all the included cells. Pure DNA is obtained in 30 minutes. 4 application-specific kits are available. The Fast DNA Spin Kit for soil, a gold standard method for the isolation of DNA from environmental samples, which is produced in a 2 ml tube version for small amounts of samples, and the 50 ml tube size kit. The Fast DNA 50 ml Spin Kit for soil that allows to process up to 10 grams of soil. Another kit, the Fast DNA Spin Kit for feces, is dedicated to stool samples, and the standard Fast DNA Spin Kit is designed for animal and plant tissues, as well as bacteria, yeast, and fungal cells in cultures. The Fast DNA Spin Kit for soil is listed in over 11,000 articles and is described by several research teams as the most efficient method for DNA isolation from environmental samples. Lysing Matrix E-tubes included in the kit ensure an efficient lysis of microorganisms in a few seconds and the complete purification process performed in 30 minutes. That means a huge saving of time compared to manual mechanical methods. Any cells included in the sample is efficiently lysed, even the most resistant ones. Gram-positive and negative bacteria, fungi, yeast, bacterial and fungal spores, protozoa, viruses, as well as plant and animal cells. High yields of high-quality DNA is purified with this method, with the guarantee of an efficient removal of humic acids and all other PCR inhibitors included in the sample. DNA is ready to use in any downstream application like qPCR or NGS, for example. Results are highly reproducible with no risk of cross-contamination thanks to single-use Lysing Matrix tubes with highly controlled size and quantity of Lysing Matrix beads. On this slide, you can see all the sample types listed in scientific papers reporting the use of the Fast DNA Spin Kit for soil. Environmental specimens like soil, sludge, sediments, or water samples, plant tissues like leaves, roots, or seeds, for example, but also food samples and human and animal tissues. I will now present you some case studies reporting the use of the Fast DNA Spin Kit for soil in various applications. The Fast DNA Spin Kit for soil, in combination with another kit from MP Biomedicals, the RNA kit, was used successfully to co-extract DNA and RNA from soil samples. The RNA-DNA ratio is indeed an important indicator of the metabolic status of microbial communities. Such a ratio can allow researchers to address questions concerning whether the response of a microbial community to environmental changes is due to a population increase or activity increase. Here, the co-extraction protocol was optimized on a sandy clay loam soil sample. Up to 26 micrograms of DNA and 4.5 micrograms of RNA per gram of soil were obtained with a very good repeatability. The protocol tested on several tropical soil samples was found to be reliable and efficient on all types of samples. In another study, water lily leaves were employed as a study model to investigate the relationship between bacterial microbiomes, biodegradation, and fossilization. Here, the research team compared 4 DNA extraction kits to identify the most suitable kit for the extraction of DNA from bacteria associated with biofilms on decaying water lily leaves for 16S rRNA amplicon analysis. The fast DNA spin kit for soil resulted in high DNA concentration with better quality and yielded the most accurate depiction of the bacterial community as shown in yellow here. The next study is dedicated to the evaluation of different protocols for bacterial DNA extraction from human fecal samples. DNA extraction from feces represents the essential step in obtaining good quality DNA and ensuring accurate identification of microbial composition and relative abundance. Comparison of 3 different methods pointed out that the fast DNA spin kit for feces yielded 3 times more DNA than the 2 other kits and achieved the best purity level. The fast DNA spin kit for feces was associated with higher richness of isolated taxa observed in alpha diversity analysis. This is probably due to its effective gram-positive cell wall disruption, resulting in extracting more gram-positive bacterial DNA than the 2 other kits. The fast DNA spin kit for soil was also used in DNA-based ecological studies in aquatic ecosystems. Advances in high-throughput sequencing technologies allow a complete study of microbial plankton community composition and diversity. The DNA extraction of plankton is a key step for such studies. The research team here evaluated the influence of 5 common commercial kits on DNA quality, microbial community diversity, and composition of plankton in the surface water of a reservoir, and showed that the fast DNA spin kit for soil produces the highest alpha diversity for bacteria and allows as well to obtain high quality and yields of DNA. Therefore, it is the most suitable method for bacterial plankton diversity study. Strengthened by this success, MP Biomedicals R&D team developed new kits named SpinEasy DNA Kit. SpinEasy kits offer the ultimate of convenience and ease, as the DNA purification process is based on a bind wash elute protocol using ready-to-use silica membrane spin columns. These kits include as well specialized Lysing Matrix tubes for the initial grinding of the sample, and the complete DNA isolation is achieved in 30 minutes. For microbiome research application, 3 SpinEasy DNA kits were developed, 1 kit for soil and other environmental samples, the SpinEasy DNA kit for soil, 1 kit for fecal samples, and more recently, a new kit for the isolation of DNA from water samples, the SpinEasy DNA kit for water. The SpinEasy DNA kit for water is designed to quickly isolate DNA from microorganisms included in water samples. Water is first filtered with the 0.22 micrometer filter membrane included in the kit. Microorganisms are retained on the filter that is then placed in the supplied Lysing Matrix E tubes and processed with the FastPrep instrument. After this lysis step of the microorganism, DNA is purified with the ready-to-use silica spin columns. The SpinEasy DNA kit for water contains a pack of sterile filters made of cellulose that allow a fast filtration of large volumes of water sample and are designed to absorb efficiently microorganisms. The 0.22 micrometer pore size filter ensures the collection of any type of microbes, and the diameter of 47 mm is compatible with commonly used vacuum filters. After the assembly of the vacuum filter set, large volumes of water can be filtered. Using forceps, the membrane containing the microorganism is then removed and rolled up into a cylinder, which is then placed into the Lysing Matrix E-tube. After the addition of 2 lysis buffers and RNase A to the Lysing Matrix E-tube, samples are processed with the FastPrep instrument to lyse microorganisms. 250 microliter of an inhibitor removal buffer is then added to the homogenate, and tubes are mixed and centrifuged to pellet the precipitate as well as cell debris. The supernatant is then collected and added to the ready-to-use spin columns together with a binding buffer. After 2 washing steps, pure DNA is eluted with the DES buffer. In the following experiment, DNA was extracted from different types of water samples with the SpinEasy DNA kit for water. Results demonstrate that high yields of pure DNA are extracted and DNA is suitable for PCR amplification. MP Biomedicals R&D team is also trending his studies toward automation. A complete portfolio of magnetic bead-based kits for nucleic acid purification was launched this year together with an automated purification platform. MagBead DNA kits are using magnetic bead with high binding capacity and selectivity for genomic DNA in order to isolate nucleic acids. They include as well specialized Lysing Matrix tubes for a complete and efficient grinding of the sample and cell lysis before proceeding with the purification of the DNA. Purified DNA is ready to use for downstream applications, including sequencing experiments. The way to use MagBead kits is very flexible. These kits can be used manually with magnetic wax, and MP Biomedicals supplies magnetic wax for 8 and 24 samples. Ready-to-use MagBead FastDNA kits are designed for the use also with the new automated purification MPure32 platform, and MagBead's FastDNA kits are also suitable for use with any other open automated purification platform, like the Kingfisher Flex, for example. You can see on this slide the new MPure32 instrument. It is one of the smallest systems on the market, 38 cm per 35 cm on the benchtop. It is easy to use thanks to the touchscreen with user-friendly icons containing up to 100 preload programs. Ultra-pure DNA and RNA is isolated from 32 samples in less than 1 hour in a reproducible way without the intervention of the user. 2 heating blocks ensure the assay to be run at the optimal temperature for maximum yield and results consistency. After use, the building UV light decontaminates the instrument. The first kit that was launched is designed for the purification of DNA from soil and other environmental samples. The use of high DNA binding magnetic beads and extraction buffers specially developed to eliminate humic acids and other PCR inhibitors allow to get high yields of high quality DNA from soil, sediment, sludge composed of manure. Extraction is performed manually or with an automated purification instrument in a process that takes 1 hour. Similarly, the MagBead's FastDNA kit for feces allows to extract high yields of pure DNA from human and animal fecal samples. Again, the purification process is performed manually or with an automated system in as little as 1 hour. No phenochloroform or other toxic chemicals are required. In order to offer the ultimate inconvenience, MP Biomedicals produces MagBead kits composed of 96 well plates, pre-filled with reagents and ready to use with the MPure32 instruments. The protocol of MagBead's FastDNA kit starts with the grinding step of the starting samples performed by bead beating with the Lysing Matrix tubes and lysis buffer included in the kit in combination with the FastPrep instrument. After centrifugation to pellet the beads and cell debris, the supernatant is collected and the purification of the DNA is performed by a bind-wash elute process using magnetic beads that can be automated with the MPure 32 instrument or another similar instrument. Gel pictures of DNA isolated from soil and fecal samples with the MagBeads FastDNA spin kit for soil and MagBeads FastDNA kit for feces show high yields of high-quality DNA. As a summary, we can claim that MP Biomedicals offers a complete solution for microbiome research sample preparation, a comprehensive range of FastPrep bead beating instruments and Lysing Matrix tubes, a large portfolio of kits for the purification of DNA, as well as an automated instrument and combined kits designed for high throughput application, answer the need of any microbiome research project. MP Biomedicals is pleased to offer you free sample kits of FastDNA spin kits, Spin Easy kits, as well as MagBeads kits if you are interested in testing one of these protocols for the isolation of DNA with your starting sample. A demonstration of the FastPrep and MPure 32 instrument can also be organized in your lab. For more technical information, detailed brochures, application notes, and video are also available on request. I would like to thank you for your attention and please contact me if you have any question.